In this lesson, we'll be talking about the Kirby Bauer antibiotic susceptibility test. There are several different types of antibiotic susceptibility tests. This one in particular is a way that we can test a single bacterium against various different antibiotics by exposing the bacteria in a dish to those antibiotics and seeing if the antibiotics are able to inhibit bacterial growth. So at one point, you've probably all used antibiotics. So we know antibiotics are a type of antimicrobial drug, meaning they're a medication that's used to destroy a microorganism. Specifically with antibiotics, they're used to cure bacterial infections. So the way antibiotics work is they typically target part of a bacterial cell, such as their cell wall, or bacterial ribosomes, or DNA or RNA or the cell membrane, and by targeting that particular part of the bacterial cell, it inevitably leads to bacterial cell death. Antibiotics typically are made by microorganisms, so there are various different species of bacteria like Streptomyces and fungi like the penicillium mold that are responsible for producing the chemicals that we know as antibiotics that can kill bacteria. So the penicillium mold is responsible for producing the antibiotic that we know as penicillin. The Streptomyces bacterium is responsible for producing the antibiotic streptomycin. For these bacteria that produce these chemicals, it's a defense mechanism to them so that bacteria don't grow in their space and use their nutrients. It's just we have used these chemicals, we've extracted these chemicals for use to cure bacterial infections in the medical industry. We can classify antibiotics based on their spectrum of activity. So with broad spectrum antibiotics, these are antibiotics that target a wide range of bacteria. So they can typically target both gram-positive and gram-negative bacteria. They typically target part of a cell that all bacterial cells have and have very similar forms of. So they're able to target this wide range of bacteria. So for example, tetracycline works by targeting bacterial ribosomes. All bacteria have ribosomes, so unless the bacteria has acquired resistance, it typically would be highly susceptible to something like tetracycline. The bad thing about broad-spectrum antibiotics is they are more prone to contributing to superinfections, which are infections that happen after taking a broad-spectrum antibiotic when the normal biota has been destroyed. It kind of leaves the space and nutrients behind for dangerous pathogenic bacteria like C. diff to grow out of control. And then broad-spectrum antibiotics are also more prone to contributing to resistant strains of bacteria. So they're highly effective, but they can be problematic. Narrow-spectrum antibiotics, they target a specific type of bacteria. So in order to effectively use a narrow-spectrum antibiotic, you have to know what bacteria you're infected with to make sure you prescribe the appropriate one. A lot of times broad-spectrum antibiotics are used when the exact causative agent isn't known, and it doesn't necessarily need to be known because broad-spectrum antibiotics can target a wide range. But as far as a narrow-spectrum drug, it would just target a specific type of bacteria. So for example, isoniazid is an antibiotic that's used to specifically target Mycobacterium tuberculosis, which causes TB. So you'd have to actively be infected with that bacteria for that antibiotic to be effective. So what we typically do is we test to see if an antibiotic is going to be effective at curing an infection. Now, a lot of times doctors and nurse practitioners and PAs will know from experience what the causative agent of an infection is and what antibiotic works best, so they're not necessarily always running to the back and doing a Kirby Bauer test. But in situations where it is unknown what the best antibiotic choice would be, the Kirby Bauer test would be highly beneficial. So typically what's done is you have to have a specific type of plate. The specific plate that you need is called a Mueller-Hinton-Agar plate. And in that Mueller-Hinton-Agar plate, you'll want to add the bacteria in a way that you're creating a bacterial lawn, which is kind of what we see here. A bacterial lawn basically just means you're covering every square inch of that plate with bacteria. It's kind of the opposite of doing a street plate technique. When you do a street plate technique, you want it to be nice and diluted so you get those isolated colonies. Not with a Kirby Bauer plate. With a Kirby Bauer plate, when you inoculate the bacteria onto the plate, you want to make sure you are thorough and that every square inch of that plate makes contact with bacteria, and bacteria have the ability to grow. Because in the end of doing the Kirby Bauer, what we do is we measure these clear areas around the antibiotic to see if the antibiotic was effective or not. But let's imagine for a second that you forgot to put bacteria in this whole section. You just missed that section when you were creating your lawn. And then you put an antibiotic right there in the middle. And then when you look at it, you think, wow, that antibiotic did a great job at killing all this bacteria. Well, really, you never put any bacteria there in the first place to know if the antibiotic would work. So when we do that, we could potentially make a kind of a false assumption that that antibiotic works when really we don't know if it does. So you have to create this bacterial lawn prior to putting antibiotics on the plate so we get like a full look at if the antibiotic is actually going to be effective or not. So typically what we do is we create the bacterial lawn and get bacteria all over the surface of the plate. Then we put antibiotics on the plate. Typically in our labs we just use 4, but you could certainly put more than 4 on these plates. They do just have to be a certain distance apart from each other because we don't want them to the medications to blend in. But you put antibiotics on the plate and then you incubate typically for 24 hours. After 24 hours, you need to look at the plate and look at the clear zones that are forming around the plate. These clear zones are very important. These are called the zones of inhibition. And this is where this antibiotic has inhibited this bacteria from growing. So really, when you see these nice, big, clear zones of inhibition, it's a pretty good sign that the antibiotic was effective. We can't say for sure. We do have to measure them and look at a chart, but it is a pretty good indication. Now, when you see very small zones of inhibition, like what we see here, or certainly no zone of inhibition, that's pretty bad. That means that this bacteria, whatever it is likely highly resistant to this antibiotic and still able to grow in the presence of the antibiotic. So a big zone of inhibition, that's good. That means the bacteria is probably susceptible or sensitive to the antibiotic. Little or no zone of inhibition, that's bad. That means the bacteria are probably highly resistant to the antibiotic. We do have what we call an intermediate range, which means the antibiotic can be effective, but typically it would have to be prescribed at higher dosages over a longer period of time. So while I said a big zone of inhibition is a good sign, you can't say with 100% certainty that it works without measuring it and looking at a standardized chart. So to measure the zone of inhibition, you measure all the way across, so the whole entire clear area all the way across in millimeters. So it always needs to be measured all the way across in millimeters. Once you have that measurement, you look to your standardized chart to see if the antibiotic was effective or not. So this is the standardized chart that we'll be using. So let's say you were looking at this and you measured tetracycline all the way across. So you measure this whole entire circumference here. And then we go to our chart and we find the row for tetracycline, which is this row right here. So let's say that zone of inhibition for tetracycline, let's say it was 23 millimeters. So we look across for tetracycline and we see where 23 millimeters would fall. Well, it's not equal to or less than 14. It's not between 15 and 18, but it is greater than 19. So at 23 millimeters, that tells us that that bacteria are susceptible to tetracycline. So that's an effective antibiotic. It would in fact work. Let's say that that zone of inhibition though was 10. Well, if it was only 10, that is less than 14. So that would tell us that that bacteria is resistant to tetracycline. So tetracycline would not be an effective antibiotic to be prescribed. Let's say it was 17. Well, if it was 17, that would put us here in the intermediate range. So it could be prescribed. Ideally, it wouldn't be prescribed unless a person had like allergies or had an allergic reaction to a better medication or was on some type of medication that would cross-react with another better antibiotic. So it would only be prescribed as a kind of emergency basis, and it would likely need to be prescribed at a higher dosage over a longer period of time. So it's very easy to use this chart. You just measure your zone of inhibition, find the specific row for the antibiotic that you're looking for, and then see if it falls in the resistant, intermediate, or susceptible category. This image is just a really good depiction of all the different ways that antibiotics work. You'll be learning about antibiotics and their mode of action in lecture, but this kind of does a brief summary. So for example, we don't get to do all these antibiotics, but I tried to pick one antibiotic from each class so you do get some exposure. Tetracycline, it works by inhibiting protein synthesis, by inhibiting the 30S or the small ribosomal subunit. Something like trimethoprim is going to inhibit folic acid metabolism. Something like rifapin is going to inhibit transcription through the inhibition of RNA polymerase in the synthesis of mRNA. So this kind of goes through and just shows you all the different modes of actions that antibiotics can take, which are basically how antibiotics can effectively get in and destroy bacterial cells. Now, sometimes antibiotics don't work. I think we all have experienced that at one point, and this can be due to drug resistance. So unfortunately, bacteria are very smart, and over time, they have acquired mutations or extra genetic information that has allowed them to learn how to survive in the presence of antibiotics. And when they're able to survive in the presence of a certain antibiotic, we say that they are resistant to the antibiotic. There are a ton of different ways that a bacterium can learn to become resistant. I've just put a few here. So for example, a lot of antibiotics work by targeting things that are inside the cell, like DNA or RNA or ribosomes. So some bacteria simply just form protective barriers that prevent the medication from getting in, which can give them resistance. So a lot of bacteria can produce enzymes that can deactivate the antibiotic. So for example, some bacteria produce an enzyme called beta-lactamase, which can deactivate penicillin and prevent penicillin from working. Some bacteria change the target of the antibiotic. So for example, antibiotics like tetracycline, streptomycin, erythromycin, they all target bacterial ribosomes. So what bacteria can do is change the shape of their ribosomes so that those medications don't recognize their ribosomes anymore. And then a lot of bacteria have something called an efflux mechanism, which basically is a pump in their cell membrane that can pump the medication out of the cell. So if something like tetracycline gets into the cell, before it can destroy the ribosome, the bacterial cell will send that medication right back out of the cell through this efflux mechanism. So it basically just pumps it right out through the cell membrane. So these are all kind of, and there's more than this, but these are some standard ways that bacteria have evolved to learn to survive in the presence of antibiotics over time. So to do a Kirby Bauer test, it is quite a simple test. You first have your uninoculated Mueller-Hinton agar plate, which you will need to inoculate with a bacterial lawn. So you'll take a particular species of bacteria and you will create a bacterial lawn all over the entire surface of the Mueller-Hinton agar plate. Then you'll want to add your antibiotics. This is typically just done with tweezers. We have these little paper antibiotic discs that have medication infused within the disc. And when you put it onto the plate, the medication diffuses into the agar and hopefully is able to prevent the bacteria from growing. But you'll add the antibiotics, and at this point you would incubate the plate for 24 hours. After incubation, so after 24 hours, you get the plate back out and you start measuring those zones of inhibition, so that entire clear area all the way across. After you've measured those zones of inhibition, that is when you'll look at that standardized chart and try to decipher as to whether the bacteria are susceptible, intermediate, or resistant as far as the impact of the antibiotic.